Welcome to Chapter 6.5 on neurons and synapses, all part of the nervous system. Okay, now when we talk about your nervous system, okay, we can separate those that nervous system into two parts. Okay, so we have the central nervous system, or CNS. Okay, and that really just includes the brain and spinal cord. Okay, so this is what's receiving and interpreting the messages. Okay, that all come from another part called the PNS, or peripheral nervous system. Okay, and these include everything except the brain and spinal cord. Okay, so all of these nerves out here that are either receiving messages and taking them to the brain, like this way, or carrying messages from the brain, so going this way, to initiate a response. Okay, so let's take a moment to draw the basic structure of a motor neuron. So I usually start out with this part in the middle here. This is called the axon. Okay, it's like a long skinny part that carries the impulse. Okay, and then it ends with these little finger-like projections that will then attach to the next neuron out here somewhere. Okay, and then we have the cell body, and the cell body is covered in these finger-like projections. And of course, there is a nucleus present in this cell also. Okay, so let's talk about these various parts of the cell. All right, so again, we have the easiest to find, the nucleus. This is called the cell body. And these little finger-like projections are what we call dendrites. Okay? And dendrites are going to receive a nervous impulse from another nerve, okay, and kind of collect it. So these are collecting little bodies here. Then we have this long, skinny portion of the nerve, and this is called the axon. And the axon ends in these little finger-like projections called axon terminals. Okay, now we want to draw what's called a myelinated neuron. Okay, so a myelinated neuron is going to have some additional specialized features. It's going to have these cells that kind of line the axon. There's some gaps in between them though. Okay, so cells that line the axon. Okay, and realistically, they really kind of wrap all the way around this axon here. So they kind of go all the way around the outside. And these are called Schwann cells. And together, they make what's called the myelin sheath. Okay, so myelin sheath. And this sheath is literally a protective layer of fatty cells around the axon here. Now, you're going to notice that there are small gaps in between these. Okay, these gaps are what we call the nodes of Ranvier. Okay, and we're going to notice that some specialized things happen in these nodes later on when we get to how these motor neurons actually work. Now, as part of a good drawing of a motor neuron, you always want to include an arrow that indicates the direction of what we call an action potential or a nerve impulse. And that is going to occur in this direction. So we're going to be going down, okay, from the dendrites through the axon, okay, down to these axon terminals, okay, and this is what we would call the impulse or action potential. Now, Some of these neurons can be teeny, teeny, tiny. Some of them can be really huge. Okay, the longest one is over 3 feet long that can run from your spine all the way down your foot, a single long, skinny cell, but always starting with the dendrites, going through the cell body, down the axon, and ending at the axon terminals. Okay, so again, just another look at how these nerve impulses travel. We're going to start at the dendrite. They're receiving messages, okay? So that's where this impulse is going to start. It's going to travel along an axon. It's going to reach the axon terminal, and then that message is going to get passed off to the next neuron. Okay, so as we start talking about nerve impulses, you're going to hear me say this word potential a lot, okay? And so batteries and nerves kind of work in a very similar fashion, okay? When we say potential, what we mean is that there's some kind of a membrane, okay? And there's a difference in charge on either side of the membrane. So in order to have some kind of a potential, we have to have a difference in charge on either side of the membrane, and these charges are prevented from just diffusing across and neutralizing. So nerve cells create a potential where it's negative on one side and positive on the other by using that sodium-potassium pump. So they're going to be pumping actively sodium ions out of the cell and potassium ions into the cell. And that's going to help to create these areas of built up opposite charges. Okay, so let's talk about what's happening in our nerve cells. So here, what we're looking at is the inside of a nerve cell. So somewhere in that long, skinny axon, okay? And here would be the outside. Now, inside of this axon, there is a bunch of negative chloride ions. So we don't normally draw them in there because they're present and they're not moving. So that's kind of boring. Okay, so what's going to happen is that this sodium-potassium pump, okay, this membrane protein, is going to actively pump out 3 sodium ions and pump in 2 potassium ions. Okay, so even though they're both positive, because I'm losing 3 and I'm only gaining 2, that means overall the inside of my cell here is going to be negative. Okay, so remember, I was starting with a bunch of these negative ions. I lost 3 positives and I only gained 2. So that means overall the inside of my cell is going to be negative. And all of that happens because we are actively pumping these ions out using the sodium-potassium pump. Now, that condition that we just mentioned is what we call a resting potential, okay? So this is when a neuron is not currently sending an impulse, okay? We've already pumped those 3 sodium ions out and the 2 potassium ions in, and that's what we call resting potential. Now, just like what we talked about, okay, the inside of the cell is negative compared to the outside, okay? So we've got a situation like this going on at resting potential. Now, how did we get that resting potential? Well, that's because we pumped the sodium ions out, so they're on the outside, and those potassium ions are on the inside. Okay, and we can kind of see that here in this picture. Okay, all these sodium ions are on the outside of the cell. Okay, and the potassium ions are these green things here on the inside. Okay, so at resting potential, sodium on the outside, potassium on the inside, and the inside of the cell is negative. All of that, again, is due to that sodium-potassium pump, okay? And so that's how it's remaining or maintaining resting potential, okay? That pump is making sure that sodium is on the outside, potassium is on the inside. Okay, so let's say that this is an axon. So this long skinny part in here is an axon. Embedded within that membrane, I'm going to find a few different things. I'm going to find the sodium-potassium pump that we talked about. Okay, I'm also going to find some ion channels for sodium and for potassium. So these are for active transport, these pumps. These are part of passive transport, okay, for facilitated diffusion. During a resting potential, okay, these guys are going to be closed. Okay, so these channels are going to be closed, and we'll tell you why in just a moment. Okay, so what's going to happen is that I'm going to take the sodium ions and I'm going to be pumping them out. Okay, so that's going to allow me to move 1, 2, 3 of them out and 2 potassiums in. Okay, 1, 2, 3 of them out, 2 potassiums in. Okay, so I'll show you what that looks like so that you can draw it. Okay, so I would start with a drawing with something like this. Okay, and when we add ATP, so I want to make sure that I put that there because this is an active process. Okay, I would start with it like this. Okay, and then again, for every one, well, let's see, for every 1, 2, 3 sodiums that I move out, I can move 2 potassiums in. 1, 2, 3 sodiums out, 2 potassiums in. Okay, so by the time I'm done with this pump, okay, this is what it looks like, where all of the sodium is on the outside and all of the potassium is on the inside. So, well, not all of it, but when we're talking about concentration. So I've got sodium on the outside, potassium on the inside, okay, and because now I have a difference in the number of ions that I moved, that means the inside of my cell is going to be negatively charged. So this is at resting potential. This is how our neurons look when they are not transmitting an impulse. Okay, now when an impulse begins, okay, we call that an action potential. So not a resting potential, but an action potential. Something is happening. And this is what we call a self-propagating wave. So self-propagating meaning kind of like dominoes, once you get them started, they just keep falling on their own without any further input, okay? And that wave is due to ion movements in and out of the membrane. Okay, and so when we say ion movements, we're really going to be able to trace those sodium ions going in and out of the cell, creating this wave-like action down the axon. So here's how that works, okay? First, we're going to get some kind of a physical stimulus to cause an action potential to begin, okay? Now remember, those sodium ions have already been pumped out of the axon, okay, for the resting potential. And what's going to happen is that physical stimulus is going to cause a sodium ion channel to open. Well, since that channel is open and since we have a difference in concentration, this sodium is going to passively diffuse back into the axon. So we had a high concentration outside of the cell, but the channel was closed. Once the channel opens, that sodium ion those sodium ions are going to rush into the cell, okay? So again, sodium ions go into the cell via facilitated diffusion. When I'm taking a bunch of positive ions and I'm sticking them inside the cell, the inside that used to be negative is now going to be positive, okay? That sudden change to a positive environment is going to cause the further channels to open. They're what we call voltage-gated ion channels. So that change in voltage, that change in charge, is going to cause other channels to open. This is going to continue down the axon. Okay, now when an action potential is traveling down an axon, that's what we call depolarization. So the inside of the cell used to be negative. Okay, we added all those sodium ions, now it's positive, so we have depolarized it, we've changed it. Okay, so remember, okay, at resting potential, my sodium ions are on the outside, my potassium ions are on the inside. Okay, so what's going to happen during a depolarization event, okay, is that the sodium ion channel is going to open. That's going to cause these sodiums to rush into the cell, okay? So I would draw it like this before, and then let's take a look at what happens after. Okay, so once this sodium comes into the cell, that sudden change to a positive charge on the inside of the cell is going to cause this potassium ion channel to open. Once the potassium ion channel opens, okay, we're going to move these potassiums from an area of high potassium concentration to low potassium concentration. So these ions are going to switch. Okay, so they're going to switch places. Now, we've still gone from a negative environment on the inside to a positive, okay? And all that happened through this changing of channels, okay? So now the sodium ions that were over out here on this side are going to come in, okay? And the potassium ions are going to come out. Okay, so during an action potential, okay, that again causes the inside to become so positive that those potassium ion channels open as well. So the potassium ions move back out to where they're less concentrated on the outside. So overall, that's going to get us that return that we want, okay? That return to that negative charge on the inside. Okay, so let me just do a quick diagram here. So resting potential, we started with sodium on the outside, potassium on the inside. During depolarization, sodium comes in, but that change to a positive environment causes the potassium ions to, ion channels to open up, and potassium goes out. So when the potassium leaves, we end up with a negative back on the inside again, and that's what we want, right? Positive on the outside, negative on the inside. But the problem is that we now have things in the wrong space. So potassium's on the outside and sodium's on the inside. That's not what we want. So to return things to resting potential, we then have to use our sodium-potassium pump again to return the sodiums to the outside and potassiums to the inside. Okay, so when we're ready to do that, again, okay, after depolarization, the sodiums have been transferred inside, and the potassiums are on the outside. In order to get this cell back to its resting potential, these channels need to close, okay? I'm again gonna add ATP to my sodium-potassium pump. That's going to allow me to pump 1, 2, 3 sodiums out and 2 potassiums back in. Okay, so again, 1, 2, 3 sodiums out, 2 potassiums back in. Okay, and now I've reestablished all of my sodiums on the outside, my potassiums on the inside, and overall my cell has a negative charge in the middle. Okay, so here's a great graphic of what's going on, right? When we start at resting potential, my cell's positive on the outside, negative on the inside. Okay, when those sodium ions come into the cell, okay, the inside is going to turn temporarily positive. When they leave, okay, we're going to return to that negative state, and this wave of positivity is going to travel down the axon. Okay, so let's take a look at this graph over here. What we've got on the y-axis tells us a lot about our electrical potential. Okay, so going up would mean more positive, going down would mean more negative. All right, now at A, this is again what we call resting potential. And this is an electrical voltage graph of the inside of the axon. So maybe that's important to note, inside of the axon. Okay, at point B, okay, what we have are those sodium ions rushing into the cell. Okay, so those sodium ions are coming into the cell. They reach a peak positivity. This is my action potential. Okay, that change to a positive charge on the inside of the axon causes these potassium ions or those ion channels to open, and this causes the potassium ions to diffuse out of the cell. Okay, so when those positive ions are leaving the cell, again, if I'm losing positive ions, my charge is going to be going back down. Okay, now at this point, again, now the sodium is on the inside and the potassium is on the outside. That's the opposite that it should be. Okay, so I have to have the sodium potassium pump repolarize. Be helpful if I could actually write this. Repolarize the cell. Okay, and then that gets us back to point A, which we already labeled means resting potential. So here we're back at this resting potential. So negative to positive, back to negative, but not negative the way we want to be. So now we've got to return things to the resting potential. Okay, so again, here's my dendrite, my axon terminal. As that impulse travels down, the action potential, that positive charge on the inside of the axon, is going to cause like a domino effect here and cause those channels further down to open up. And that wave is going to travel from the dendrites down to the axon terminals. And that, of course, is all due to the movement of ions. Okay, so here are my sodium ions, these blue ones. When they come in, that gives me a change to a positive charge. Okay, that causes the sodium or potassium ion channels to open up, and I'm going to return to a negative charge. And then when I'm at resting potential, sodium should be outside, potassium on the inside. Okay, and I should be noticing a positive charge on the inside of my cell. Now, earlier when we drew our neuron, we drew a myelinated neuron. Okay, so a myelinated neuron means I've got the dendrites, got the axon terminals, I've got the axon, but all along that axon, I've got these Schwann cells, which make up the myelin sheath. So when you're drawing a myelinated neuron, you should be able to just draw it like we drew it before. Okay. If you're drawing a non-myelinated neuron, you're going to have your dendrites, really bad ones. You're going to have your axon. You're going to have your axon terminals, okay? But on your axon, you're not going to have any of the Schwann cells. So this would be a non-myelinated neuron here. So again, we've got the dendrites, the nucleus, the cell body, the axon, and the axon terminal. But we don't have any of these Schwann cells to make up the myelin sheath like a myelinated neuron would. These myelinated neurons, okay, are kind of indicative of a much more advanced nervous system, and that has to do with the way that the nerve impulses travel through these nodes, these gaps, which we labeled previously called nodes of Ranvier. Okay, so a regular action potential, like what we've been looking at, travels straight down the axon. Sodium in, sodium out, sodium in, sodium out, all the way down, all the way down, all the way down. Remember that anytime we have to return to resting potential, we've got to be pumping that sodium out. This is not only going to take energy, but it also takes time, right? Sodium in, sodium out, sodium in, sodium out, da 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 da. Okay, myelinated neurons are able to conduct what we call saltatory conduction. And instead of that happening all the way down the axon, in and out, in and out, in and out, like what we talked about normally, this is only going to do that in and out thing at these gaps, these things called nodes of Ranvier, because that charge isn't going to be able to just travel down through the axon to the next gap where it can go in and out, okay, to create that action potential, okay, and that's because these Schwann cells on the outside are insulating, okay, they're preventing those charges from leaking out, okay, so they just travel straight down here, okay, it's a lot faster and a lot easier, okay, it means that, again, instead of going in and out and in and out and in and out, okay, I can just jump from node to node to node to node to node, okay? Saltatory conduction. Now, what happens when we make it to the end of one of those axon terminals? Well, then we get to a problem, right? So this nervous impulse has to be able to jump from the axon terminal of one neuron to the dendrite of another across a space that we call a synapse, okay? So a synapse is the place where the terminal of one axon meets the dendrite of another neuron. So an easy way to kind of draw that is like this. Okay, where here we have the axon terminal of one neuron and the dendrite of another neuron. So the axon terminal would be the end, the dendrite would be the beginning of the other one, and this space in between is what we call a synapse. Okay, so the synapse is the space. And so sometimes we'll refer to this as the presynaptic neuron, pre meaning before, and the postsynaptic neuron. So a nerve impulse has to travel down the presynaptic neuron, cut across the synapse, and then get captured and travel down the postsynaptic neuron. Okay, so this is going to happen in a series of somewhat complicated steps. So first things first, the action potential reaches the terminal end of the presynaptic neuron. So that's that sodium moving in and out, the potassium moving in and out, etc. So that's going to reach, the action potential is going to reach, okay, the end of that neuron. That action potential, so that positive nature inside the axon, is going to cause calcium protein channels to open. So outside of our nerve cells, there's an abundance of calcium. So we have a high concentration on the inside and a high concentration on the outside, low concentration on the inside. But ordinarily, the protein channels that let them through are closed. Well, this positivity from the action potential causes these guys to open. So calcium is going to diffuse into the cell. On the inside of the cell are vesicles, okay, little sacs made out of the same material as the cell membrane, little vesicles containing something called a neurotransmitter, okay? Neurotransmitters are what are carrying the message of the impulse, okay? So they're sitting in there ready to go. The calcium ions cause these vesicles to push up against the cell membrane. When those vesicles fuse with the cell membranes, that's exocytosis, and all of those little neurotransmitters are forced out. Now, here's my presynaptic neuron, here's my postsynaptic neuron. When all of these little neurotransmitters are forced out, they're sitting right on the other side just like this. So here's a high concentration of neurotransmitters. Here's a low concentration. These guys are literally going to diffuse this way, okay? So here we've already talked about a couple forms of transport. This exocytosis process is active. This diffusion process is passive. All right, on the other side, on this postsynaptic neuron, there are some binding sites, okay? So membrane proteins, one of their functions can be binding sites, and these little neurotransmitters are going to bind with these proteins after they diffuse across the synaptic cleft. When those neurotransmitters bind with those proteins, that binding is going to cause the sodium ion channels in this postsynaptic neuron to open, okay? That causes sodium ions to enter the cell, and that's what's starting a new action potential now in the postsynaptic neuron. So we know that action potentials have to do with the entering of sodium ions, okay? That happens because of the binding of the neurotransmitters. Now, we've got to do something with those neurotransmitters. So they're sitting here in the synaptic cleft, okay? We need to get them back into the presynaptic neuron to get ready for the next nerve impulse, okay? So there's a couple ways that can happen. So in some instances, an enzyme will come along and eat all these and make the presynaptic neuron manufacture new ones. In other cases, we're going to be able to do a reuptake pump, okay, where these neurotransmitters are, again, actively pumped back into the presynaptic neuron. So again, another example of active transport here. Okay, so just another view of what this is looking like here. My nerve impulse travels down the presynaptic neuron. That positive charge causes the calcium ion channels to open. Calcium from the outside of the cell diffuses passively into the cell, and that calcium pushes these vesicles up against the presynaptic neuron. That causes them to be released via exocytosis. Then they passively diffuse across the synaptic cleft. The neurotransmitters then bind with receptors on the postsynaptic neuron, and that starts a nerve impulse in the postsynaptic neuron. These neurotransmitters then need to get pumped back into the cell to get ready for the next impulse. Okay, so here's what that looks like, okay? An action potential is coming down the presynaptic neuron, okay? That action potential is going to cause this calcium ion channel to open, okay? And calcium is going to then diffuse, that says calcium, I swear, into the cell, okay? So now I have a bunch of calcium on the inside here. I also have vesicles full of neurotransmitters. Okay, so I'm going to label these as NTs, neurotransmitters. All right, that calcium is going to push these neurotransmitters against the cell membrane. Okay, so the calcium ions are going to push these vesicles up against the cell membrane and force the contents onto the outside of the cell. Okay? These neurotransmitters are then going to diffuse across the synaptic cleft and bind to receptors on the postsynaptic neuron. That causes an action potential to be generated, okay, and travel down the postsynaptic neuron because this sodium ion channel opens, okay? So that means then that sodium is going to enter the cell, and that's where I'm getting this action potential from. Of course, then, these neurotransmitters then need to be pumped back in here where they're wrapped up again in vesicles, okay, and getting ready for the next impulse. Okay, so we've talked about how nerve impulses travel from a presynaptic neuron to a postsynaptic neuron across the synaptic cleft. Well, what if we're talking about a motor neuron that's supposed to trigger a muscle movement? In that case, that motor neuron would end up not touching another neuron, but instead touching a muscle cell. So its axon terminals end by connecting to a muscle cell. When this neuron needs to tell this muscle cell to do something, it's going to release a specific neurotransmitter called acetylcholine. Okay, so the way that acetylcholine works is when that action potential reaches the end of the presynaptic neuron. Okay, again, those calcium ion channels open, calcium rushes in, it forces this neuroreceptor containing vesicle to fuse against the membrane, okay, and that acetylcholine neurotransmitter diffuses across and lands and binds to the receptors on the muscle cell. Well, one of the new hot insecticides that's out on the market mess with the ability of acetylcholine to bind with those receptors, and those are called neonicotinoids. Okay, so neonicotinoids permanently bind to these receptors that are on the muscle cell. So when those receptors are already full, okay, when they already have something attached to them, then these neurotransmitters won't be able to bind there because, again, they're already full of these neonicotinoids, okay? They're blocking them. So that's going to mean that regardless of what kind of signal is going down the nerve, nothing is going to happen with the muscle. It kind of, in effect, paralyzes the muscle because even though the nerve is sending the message, okay, that message is getting blocked and not reaching the muscle cells. So that means the muscle can't contract, okay, which means that that insect is going to suffer from permanent paralysis, and of course that would lead to death. Okay, so transmission of nerve impulses down an axon and across a synapse all involve massive amounts of transport. Okay, so let's quickly review the types of transport. So we have regular diffusion, which you might have heard referred to as simple diffusion. We have facilitated diffusion. Okay, we have protein pumps. We have endocytosis, and we have exocytosis. Okay, so these two are both passive. Okay, these are my three active types of transport. Okay, and your job is to go through what we talked about with the nervous system and to figure out some examples of all of these different types of transport. Okay, and that'll do it for chapter 6.5 on nerve transmissions.